The last thing that we're going to look at is how we deal with carbohydrates, getting it to the place that either we're going to fund sugar to go into glycolysis, so we're going to talk about that, or we're going to make glucose from other entities, or we're going to process glucose in a different way. And so it's kind of a varied way we're going to look at the carbohydrates, and we've got a couple segments in which we're going to really deal with it. Before we do that, let's go to the Lord in prayer. Dear Heavenly Father, I do thank you for this day. I thank you, Father, for my students. I pray, Father, that you will touch them, that you will give them the strength and the ability to look at all of this material and to be able to organize it in a way that makes sense, that puts them in relationship, that they can understand later and even further than the test. I pray, Father, that you will give them a special insight and wisdom on how to do that. I pray today that as I present the material, I present it in a way that will start anchoring some of those connections so that they can see and understand. I ask this all in Jesus' name. Amen. All right, so this chapter is a little bit all over the place, and so we'll break it down into segments. Now, we've got all sorts of things working together. We have already seen how glucose goes into the citric acid cycle, and that was glycolysis. All right. We looked at the citric acid cycle in quite a bit of detail, and that's great. But what we really haven't done, and a lot of books will introduce glycolysis together with a pathway that's called gluconeogenesis, the reverse. Okay, you've probably taken a look at that. We're going to take a look at that again in this chapter to articulate it for these contexts. So we're going to look at how we can convert things back into glucose. It is very important for us to be able to do that because glucose is used by the brain exclusively. There is a limit, though, to what we can actually derive in terms of energy. And what we can do is we can take the amino acids and put them into glucose. We can also take glycerol of the fat cells, which is only 1,3 carbon intermediate of a fat cell, change it into glucose. But what we can't do is take the fat and convert it into glucose. So our largest store of energy that we have in the body, if by chance we're running out of glucose, we can't convert that very massive store of energy into that particular form. We can do it into other forms, but we can't keep our brain alive just with our fat stores. Then from glucose 6-phosphate, we can make all sorts of things. We can make, for us, storage glycogen. We can talk about, and we will talk about how to send it off to the bloodstream for the brain. In plants, we can make starch and sucrose, which we touched on, and we'll go back over a little bit. And so we're going to look at different ways we can process glucose. The first way to process glucose is glucose 6-phosphate has the ability to go down glycolysis that we see, and that's most of the time what biochem texts talk about. But there's an alternative pathway for its degradation, and that is the pentose phosphate pathway. Now, what is the outcome of the pentose phosphate pathway? Two things. NADPH is produced, which is a source of reducing hydrides in biosynthetic pathways. And then we're going to, or can, create ribose 5-phosphate, which is the sugar base for all of the precursors that we've been working up to this point in time. And so to make our DNA, we need glucose, because that's where the sugar is going to come from. Okay? Now, the pentose phosphate pathway has the ability to make these, and if you go down the pathway a certain way, you can make both of them. But if we go through mechanism, or through pathways, we can make only one of them. We can make only NADPH exclusively without generating any ribose. Or we can make ribose exclusively without generating any NADPH. And what will happen is we're going to work in concert with 3 different pathways: glycolysis, gluconeogenesis, and the pentose phosphate pathway. And depending upon what the body needs, we will go through one or many of these to generate what the outcome we need. And so that's kind of an oddball situation. Up to this point in time, we've looked at one pathway by itself, and we've either told how do you upregulate or downregulate it. Okay, but the endpoint, the beginning and the end were kind of standard and kind of defined. The pentose phosphate pathway, on the other hand, it can go lots of different ways depending upon the conditions in the cell. This is a lot more realistic look at metabolism because certain pathways work in concert and pathways are upregulated or downregulated to keep the whole homeostasis, everything in balance. And so sometimes parts of it on, sometimes all of it's on, and it all will depend upon what the body needs. The pentose phosphate pathway is broken up into 2 portions: the oxidative portion. The outcome is ribulose 5-phosphate and NADPH. Ribulose is not ribose. It's a sugar that is related. And then there is a non-oxidative pathway that will actually produce the ribose, and then it will produce glyceraldehyde 3-phosphate and fructose 6-phosphate. At the end of the pentose phosphate pathway, the 2 molecules that are created in terms of the carbons, there and there, are actually intersecting 2 pathways, either glycolysis or gluconeogenesis. So those molecules can either be ramped up, go back to glucose, or they can be sent down and produced ATP by going through the citric acid cycle. And so we'll talk about how to get those 2 together. The oxidative portion of the pathway uses the enzymes glucose 6-phosphate dehydrogenase. Okay? And the dehydrogenase is where we're going to have hydrides flow and a redox reaction. In this particular case, what we see when we do inventory is the ring is all the same except around this carbon. Here I have a hydrogen and a hydroxide. Here I have a ketone. And so NADP plus allows us to have a hydride as a leaving group because it's going to accept. So our base will take this hydride, create that ketone, and the NADP plus is going to accept the hydride. And we have created one of the NADPHs that we can in this pathway. We have a cyclic intermediate, but if you take a look at it, we have a ester right here. And so I can react water and have it attack the carbonyl. Electrons will kick up, electrons will kick down, and then this hydroxide, oh well, let me try this again. This hydroxide will pick up a proton. So if you take a look at this, and if I label this one right here, that oxygen is red, that oxygen becomes this oxygen. This green oxygen is that one. And whatever this color is right there, I actually can't tell much of a difference, but is this oxygen right there. So you can articulate where all of that goes. And we get 6-phosphogluconolactone. All right, we need the open chain structure for the next reaction. And what we're going to do is use another dehydrogenase. And so, again, doing the inventory, hydroxide becomes a ketone. We create another NADPH. Okay? And I create an intermediate. Now, one thing about ribose and glucose is that there's a difference of a carbon. And so we are going to create CO2. And we're going to decarboxylate. Now, the oxidation step, the redox step in this previous reaction is absolutely important for the mechanistic step to create CO2. Not just that we need NADPH, which we do need NADPH, but it creates the reason why the carbon dioxide can fall off. Okay? Potentially, any acid has the ability to take one of its pairs of electrons and form a double bond. And CO2 is a wonderful leaving group. But the problem that we have is this. Most of the time, and I'm going to do it on this intermediate, which it doesn't happen, and I'll show you why. If by chance those pair of electrons fall in to make the CO2, this pair of electrons can only reside on that carbon to create a carbanion. And that's a very high energy state, and it's so high energy that it just won't happen, even though CO2 is a wonderful leaving group. But once I do the step of the oxidation, instead of the electrons sitting on that carbon, what can happen is we can create this intermediate where the negative charge is sitting on the oxygen. That is still high in energy, but it is much, much lower than a negative charge on the carbon. And so that intermediate allows the CO2 to decarboxylate. Now, we do have to regenerate. And so the next step in the mechanisms, it does reform with the double bond. And the temporary double bond that was formed picks up a proton to form ribulose 5-phosphate. Okay. Ribulose. We have now completed the part of the pentose phosphate pathway that is considered oxidative. Now, depending upon your textbook, this textbook actually identifies this as the non-oxidative step, but a lot of textbooks throw this one reaction into the oxidative process. Okay, but it's not oxidative because it has no hydride moving. The ribulose 5-phosphate and the ribose 5-phosphate, which I think you need to change the name in your notes. I had to change it in the slide this morning. Only difference here is where the hydroxide and where the ketone is. One is a ketose, one is an aldose. And so we use an isomerization. And remember, when we use an isomerase, we deprotonate and protonate a couple times until we get to where we need. And so if I take a look and I deprotonate, I'm going to deprotonate one of the protons. It will fall in, and then this will become a hydroxide. We get 2 hydroxides. And then in the second step, a base, that same enzyme will catalyze both of these movements. We'll create the isomerization. And we've got ribose 5-phosphate. So if I look at the stoichiometry right now, for every 1 glucose, what do I make? 1 glucose creates what? What was my outcome of these steps? 1 ribose 5-phosphate, right? Plus what else? What else did we make? We made CO2 plus what else? How many? How many NADPHs? 2. We actually had 2 steps that created NADPH from 1 glucose. Okay? And so 1 glucose actually derives a whole bunch of stuff. Oh, next slide. Got it all done. Okay? We did have to add some water, but water's ubiquitous. And so we get 1 ribose 5-phosphate, R5P. We're going to use a lot of acronyms, or you're going to be writing a lot. A CO2 and 2 NADPHs. So we get actually a lot of hydrides associated to that. And NADPH is going to be used to donate hydrides. But if NAD plus had accepted those electrons, we could have gotten 5 ATPs from that, from glucose. So that represents a lot of energy. And I still have a lot of carbons left in that ribose to use later. So we've got that. So I said the pentose phosphate pathway makes both of those 2 things. That is true. But there is a non-oxidative portion of the pathway that can take those carbons and do something with them. The non-oxidative pathway will take 3 of those 5 carbon intermediates. Okay. And a lot of times the stoichiometry is this. It takes 3 riboses, although I'm going to articulate that that's a little bit wrong. And 3 riboses represents 15 carbons. It will take 2 enzymes, one called transaldolase and one called transketolase, and it will do reactions with them to create outcome of a 6-carbon intermediate, a 6-carbon intermediate, and a 3-carbon intermediate. And we said it would make fructose 6-phosphate, 2 of those, and 1 glyceraldehyde 3-phosphate, 1 of those. 6 plus 6 plus 3 is 15, right? 6 plus 6 is 12, plus 3 is 15. Without your calculator, how in the world is that happening? Okay. Tell me, looking at the reactions, how many carbons does transketolase transfer from one entity to the next? Does it transfer 1? Okay. If I transferred 1, I would make this one less, and I make that one more. Okay, so I would end up with a 4 and a 6. Oh, transaldolase. Transaldolase. If I took 1 and transferred over, I take 1 over here and transfer it over here, so this would become 6 and that would become 3. That's possible. Is there any other way transaldolase could transfer something and end up with those same numbers? Yes, it can do 3. If you assume that this one goes to that one, the 7 can take 3 and give it to the 3 to make 6, and the 7 would become 4. And transaldolase moves 3. What about transketolase? It will move 2. Okay. And so 2 come from this one, are added to this one to make 7. And then this one, 2 is taken away, becomes 3. In this case, I'm going to take 2 from the 5 and create 6. And 2 away from 5 gives 3. All right, so those are the movements. Here are the specific 2 and 3 carbon intermediates, and it has to be in this order of hydroxides and ketones, and it has to be in this stereochemistry because enzymes are very specific to what they bind to. All right. So it brings those particular pieces. Now, those particular pieces are ends of sugars. Okay, so this is not a big deal in terms of we are dealing with all sugars here. And so here are some of the steps. Now, remember that I said that you make ribose at the end, right? And we always talk about pentose phosphate pathway as making ribose. Well, we don't call it the ribose phosphate pathway because we actually use a lot of 5-carbon sugars. And so the end of the oxidative gives us ribulose 5-phosphate. Okay? And that can go to, and we've seen it go to, ribose 5-phosphate. Okay? But that ribulose can also be epimerized into xylulose 5-phosphate. And we're going to need some xylulose 5-phosphate. Okay, before we get to, this is the transformation before we get to this explanation of this slide right here. Okay. So the first reaction is 1 ribose and 1 xylulose. And I'll take transketolase, and I will take 2 carbons and transfer them over. The 2 carbons come from the xylulose, and I stick them right there to make the pseudohepulose 7-phosphate. So in that other slide, we said it made a something with 3 and something with 7. All right. Not done. I now have glyceraldehyde 3-phosphate and the pseudohepulose 7-phosphate, and then I do transaldolase. And what I do is I take 3 of them that are right here, and I put them right there. And so I get fructose 6-phosphate. This is the endpoint for one of the processes, or that can be an endpoint for one of the processes. And then I got erythrose 4-phosphate. Okay, so here in stoichiometry, I have a 3, I have a 7, it goes to a 6, and a 4. 10 carbons on one side, 10 carbons on the other side. I take that 4 and I add another 5 carbon to intermediate in the form of xylulose. Okay? And so I take transketolase and I transfer 2. The 2 I transfer are these top 2, and I put it right there. I create fructose 6-phosphate, which is 6, and glyceraldehyde 3-phosphate, which is 3. 4 plus 5 is 9, 6 plus 3 is 9. I've made now 2 fructoses, fructose 6-phosphates, and 1 glyceraldehyde 3-phosphate. And that is all good. Okay? And so a lot of books will say 3 riboses will enter in the non-oxidative pathway, and I'll get glyceraldehyde 3-phosphate and 2 fructose 6-phosphate. But really, it is 2 xyluloses and 1 ribose that react, because ribose can be converted to ribulose, and ribulose can be converted to xylulose. And so you will see both of those equations in different textbooks depending upon how detailed they want to be about this process. Now, this is when perfect stoichiometry works. Okay? I bring in 15 carbons, I put out 15 carbons and convert them. In real life, does everything react with perfect stoichiometry? It does not. This is where your quiz is going to come into being. Okay? Now, there are different fates in which glucose 6-phosphate can interact with the pentose phosphate pathway depending upon what the cell needs. And so the body will send different molecules down different pathways to fulfill getting the concentration of whatever it needs up to the homeostasis point. And so I can have conditions in which I actually need the ribose, but I do not need the NADPH. Okay, well, we said to go from glucose to ribose, we're going to make NADPH if I go down the oxidative portion of the pathway. Well, sometimes ribose 5-phosphate and NADPH are both needed. Sometimes I need more NADPH than ribose 5-phosphate. And then sometimes I need NADPH and I need pyruvate so that it can be broken down to make ATP. And so there are different modes in which this can interact, and it is all because the body can have different concentrations of NADPH in the body and ribose and ATP. Now, the mode 2 is what I just showed you. Both the the mode 2 is the oxidative portion and the first non-oxidative state where I take all the glucose and I make ribose 5-phosphate and I get lots of NADPH and I lose the CO2. Okay? So that is how we traditionally look at the pentose phosphate pathway. But under these conditions, what if I just need ribose 5-phosphate, but I can't do the oxidative step because there's no NADP plus roaming around? That means I can't use those enzymes that I wanted to use. So this is what the body does. It sends glucose down glycolysis. And it makes large pools of fructose 6-phosphate and large pools of glyceraldehyde 3-phosphate. Now, if you looked at the transketolase transaldolase reactions, they were all in equilibrium. And so, if I took 2 of those and I took 1 of these, I can make 3 of those using the non-oxidative steps of transketolase transaldolase. I can make this without making any NADPH. I just have to go about it a different way. All right. Here's a different case. You do need to make a lot of NADPH, but you don't need a lot of ribose. Okay? And so the pathway to make NADPH is the oxidative step. So we go down that way. That's great. And then I can make ribose. That's great. But I don't need ribose. Well, this is what you can do. You can go down transketolase and transaldolase and make all these glycotic intermediates. And then you can go up gluconeogenesis to make more sugar back. And you know what you can do again? You can go a second time. And you know what you can do again? You can go a third time. And you can run this in a circle until you made enough NADPH that the body needs. Okay? Can you infinitely go this direction? No. Every time you run through 1 glucose, you lose a CO2. So you're eventually going to convert all your glucose into CO2 if you continually go down that circle. But yeah, you should have enough glucose 6-phosphate in the system, and your NADPH levels only have to move a certain amount that you'll get the concentration up to where you need it, and then it'll stop. The last mode, if by chance I need NADPH and I need ATP, oxidative portion of pentose phosphate pathway, non-oxidative portion of pentose phosphate pathway, and then we finish out glycolysis, which then goes to the citric acid cycle, which then goes to oxidative phosphorylation, which then goes to ATP. All right. Those are different modes by which glycolysis, gluconeogenesis, and the pentose phosphate pathway, both of the stages can work together. So, I can see a wonderful test question coming from that. I'll give you a cellular condition. You tell me what's working. Okay? And so, how do those three interact together to get the body where it needs and to get what we're actually trying to get to in terms of reactions? All right, the mechanism of transketolase I wanted to show you transfers 2 carbon units, and it uses TPP. Okay? The bigger thing when I'm looking at reactions is for you to classify and make relationships. Where have we used TPP before, or have we used TPP before? We have, haven't we? Where did we use it? Okay. Yeah, pyruvate to acetyl-CoA. So it was the step between glycolysis and citric acid cycle. Okay. So we've used that particular cofactor before. It is a derivative of the vitamin B1. Okay. And so we're using the same kind of cofactor. And so TPP, again, looks like this. And we're going to use it the same way. We're going to deprotonate, and we deprotonate right here to create a nucleophile. All right? And we've got a carbonyl. Remember, we also want to transfer this carbon and that carbon. That's the whole point of transketolase. And so that carbanion will attack that ketone, and I create an intermediate. And again, I want to carry this carbon away, and I want to carry that carbon away. I will react with a base, and I will break the bond between the entire R group, the sugar that starts off with, and we can react with a lot of sugars, and I break off and I leave 1, 2 carbons sitting on the TPP. That creates a resonance structure. Okay. The equilibrium lies way over here. The reactive species, though, is the carbanion. The carbanion then can react with an incoming other sugar that has a aldehyde, and I can create a carbon-carbon bond where I have transferred 1, 2 carbons that are sitting now on the other sugar. The TPP was just there to hold on to those 2 for a while. The last step, if I deprotonated, I reform the ketone, the TPP is regenerated, and now I've taken 2 carbons from one sugar and put it onto another sugar. And we saw 2 reactions of transketolase in our non-oxidative steps. Transaldolase is going to carry 3 carbon units. Now, transaldolase really kind of sounds like another enzyme that we've actually looked at before. What was the name of the enzyme, and what was the pathway that enzyme was in? Have you heard an enzyme that is similar? Yes, we've used aldolase. What pathway was aldolase found in? Glycolysis. And that was the one that broke the 6 carbon chain into 2 3s. All right. So basically what it did was the enzyme broke 3 pieces off and made it come off. Here, transaldolase is going to take 3 carbons off and not release it, but actually transfer it to something else. Okay? if we went back to the enzyme mechanism of aldolase, we knew that the enzyme will have a lysine that reacts to a ketone that facilitates the reaction. And in this case, they are named similarly because the enzymatic pathway is absolutely the same. There is a lysine that gets deprotonated. It will attack the carbonyl. The carbonyl will kick up and pick up a proton to become a hydroxide, and then I deprotonate the amine and the hydroxide leaves, and I have converted a carbon double bonded to an oxygen to a carbon double bonded to a nitrogen, and the important part is it is protonated. This particular organic functional group is called a Schiff's base. Schiff's bases are used all over the place. Schiff's bases hold cofactors inside enzymes. Schiff's bases hold or become part of reactions in mechanisms, and this is the second one that we see. And so the reason why a Schiff's base is used is because it connects the protein to the organic molecule covalently, and so it's held on really, really tightly. But it can be easily reversed by adding water. So it's reversible. It holds on, and then it can fall off. The reason why we have that Schiff's base is if we deprotonate a hydroxide, the leaving group is not so good with a carbanion that is seen here, but this carbanion can create a resonance structure, and this one is very, very stable. And so it stabilizes the transition state. Again, this will be the reactive species, but the equilibrium lies way on this side. I have now taken 1, 2, 3 carbons away from something that came in, and I have 3 carbons to move. I will bind to another sugar designated by R2. The carbanion will attack the ketone to create an alcohol. And then the last step, because now we've connected those 3 carbons to another sugar, is just to get rid of the lysine by adding water and regenerating the carbonyl that originally was there. And so there's the mechanism of how the transaldolase works. And so in this whole mechanism, those complex transchelase transaldolase reactions, we've already seen both cofactors, both mechanisms done in other transformations of sugar. All right. If you take a look at some stoichiometries, we showed you 3 5-carbon intermediates will go to 2 6s and a 3. Well, if you have the perfect stoichiometry, you can take 1, 2, 3, 4, 5, 6 5-carbon units. So 6 times 5 equals 30 carbons. And you will end up with 1, 2, 3, 4, 5, 6 carbon units. 5 times 6 carbons, and this is 6 times 5 carbons to make 30 carbon units. Essentially, you can say 5 riboses, 5 ribose 5 phosphates, not 5, 6 ribose 5 phosphates can be regenerated into 5 glucose 6 phosphates. And so if I take 6 of the ribose 5 phosphates, I can make 5 glucose 6 phosphate. And that just does not look like a G and a 6. All right, G, 6, P, glucose 6-phosphate. And so there's some really nice stoichiometry that can go on in this type of reaction. But if I feed in glucose and I go through the pentose phosphate pathway and I come back through gluconeogenesis, I can regenerate glucose 6-phosphate. But I lose CO2 for every one each time. So each time I go around, I can generate less glucose and less glucose and less glucose. Eventually, I will run out. Pentose phosphate pathway regulations. All right, we learn how it actually happens, and on top of that, we have to learn what turns it on and turns it off. This one seems fairly easy. There's an allosteric regulation in which the enzyme that goes down the pentose pathway, the glucose 6-phosphate dehydrogenase, the one that sends it that direction, responds to this. If there's NADPH in high concentration, it binds to the enzyme and makes it less active. And so the outcome of that is to make NADPH. If I have a lot of NADPH, it's kind of a feedback. Conversely, if there's a lot of NAD+, meaning there's a lot of reactant for that particular enzyme, it binds to it and upregulates it. So that one is a real simple, it's going to make NADPH, and NADPH is what's going to regulate it. Okay. Now, why in the world do we need to make NADPH? One way I described already is for biosynthesis. But there is another reason why we actually make NADPH, and that is to detoxify radicals or oxidative stress. And so NADPH is going to give the electrons to a molecule called glutathione. And glutathione is in high concentration inside the cell. It acts as a buffer of hydrides to react with oxidative species. And the glutathione peroxidase, okay, will react to reduce the reactive oxygen species. It will react two glutathiones to make the oxidized glutathione intermediate and donate the hydride to create the water, which is safe for us. To regenerate the reduced glutathione, our NADPH is used to make sure that there's a lot of that. And it's actually in very, very high concentration. Now, one thing that happens is that there is always oxidative stress that happens in cells. And we always have to reduce these. One place in which these radicals come from is simply the oxidative phosphorylation. Sometimes the electrons escape and react with water or oxygen and create reactive species. And so we always are having to make sure that we reduce these things. There are some people that have a deficiency in the glyceraldehyde 6-phosphate dehydrogenase, which then limits how much NADPH they can generate, which then causes issues with dealing with oxidative stress. There are a couple of things that are given in terms of drugs, like antimalarial drugs or some foods that you might eat, which tend to, when you take them, cause a little bit extra oxidative stress. And a lot of times people will find out that they have the deficiency in the enzyme because they become very sick if they eat certain foods or take certain drugs. And their dehydrogenase works just enough to keep up with biosynthesis, but it doesn't keep up enough with making enough glutathione in the reduced state, and so you can see an illness. And this is one way that we detect that particular illness. Gluconeogenesis, you actually know just about all you need to at this moment in time, because the majority of the enzymes in glycolysis are in equilibriums. So most of the enzymes in glycolysis are also the enzymes in gluconeogenesis. There are only 3 places in which glycolysis has to be bypassed. And this is the place where ATP is generated. That enzyme is a single arrow. There's a lot of energy given off, and it can't be just reversed in that particular case. And when ATP is used to phosphorylate, and where ATP is used to phosphorylate. So those 3 enzymes in glycolysis, hexokinase, the phosphofructokinase 1, and the pyruvate kinase, can't be reversed. So up at the top, very simply, a kinase puts on a phosphate. The reverse in gluconeogenesis is a phosphatase. Take off the phosphate. A kinase puts on the phosphate, easily reversed by an enzyme that takes off that phosphate. This step's a little bit more complex. It actually takes 2 reactions to bypass that one step because of how much energy it gives off. And so to get past that step, I have to invest ATP and I have to invest GTP. So 2 equivalents of ATP to get past that one step to get it back up to phosphoenol pyruvate. We'll take a look at those 2 reactions. Well, we'll take a look at all these reactions in the next couple slides. We need gluconeogenesis to run because not only the brain but also the red blood cells use glucose exclusively. The brain requires about 120 grams of glucose daily, and if you actually mobilized every bit of the glycogen stores that you have or an average person has, you only have 190 grams available. And so we have to have the ability to create glucose. And if you start going through a starvation state, it's looking at that, you have less than 2 days before your brain runs out of food, and we know that we can last longer than that in a starvation state. The precursors, there are not a lot of them, okay? The lactate that is produced in anaerobic exercise can be reconverted. That's only if you've done anaerobic exercise, and so that may or may not be available. The amino acids is probably the largest portion of carbons that can be converted into glucose. Has anybody ever watched that show on, I can't even remember, Discovery Channel, I think it is, Naked and Afraid? Okay, so they go out into the wilderness, they're dropped down naked, and they get to take one item. Usually one person takes something to light a fire, which is really good, and another person takes a knife. Okay, and so there are 2 people that have to. Originally, in the first the first season, what they would do is they would describe the people, and they would actually tell them, you know, what their weight was at the beginning, and then I think they're out there for like 21 days. They would take their weight at the end. Many of those people, even though they're survivalists, don't eat a lot. I mean, 3 bugs every week is not a whole lot of calories that they're bringing in, and sometimes it is that bad. And they will lose a ton of weight because they will be using their stores. Not only do they lose their weight, but they used to take pictures of before and after, and you would see them slim down, and the majority of the weight loss was not in the form of fat, but it was actually their proteins being broken down to convert it into glucose. And then the glycerol, the only 3 carbons of a fat, which in general has 51 carbons. The typical fat has 51 carbons. We can only use 3 of those to make glucose. The other 48 are broken down in other ways. The hydrolysis of glucose to pyruvate releases about 20 kilocalories per mole of energy. which is pretty, it's a negative 20. That's pretty good in terms of the energetics for it. And so we have to invest energy to get past that or to go in the opposite direction. Hexokinase reversed by glucose-6-phosphatase. And a phosphatase will just hydrolyze a phosphate off the entity, off, in this case, the glucose. The glucose then, once it's in the cell, can actually bind to the transporter and go from outside the cell into the bloodstream. The phosphofructokinase, and remember, if you do that, it's usually 1. Sometimes they don't put the 1, but if the 1's not there, it is understood to be 1, is bypassed by the fructose 1,6-bisphosphatase. I'm going to cleave off a phosphate. And here are the other two, pyruvate carboxylase and phosphoenol pyruvate carboxykinase. I will take pyruvate, the end, and I will convert it into a molecule called oxaloacetate. Okay, you have heard about oxaloacetate. What pathway did you associate oxaloacetate with? Citric acid cycle. Now, oxaloacetate is either citric acid cycle or gluconeogenesis. And then that oxaloacetate gets converted into phosphoenol pyruvate. The pyruvate carboxylase will actually take a carbon dioxide, and remember, why is that carbon dioxide? Why is the bicarbonate carbon dioxide? Do you remember? This is the buffer system. CO2 reacts with water to make carbonic acid, and it deprotonates into a proton and bicarbonate. So when you see bicarbonate, it can be equated in your mind as CO2 because those are in equilibrium all the time. And so bicarbonate will provide a CO2 onto a cofactor called biotin. It's also called vitamin H. And it will carboxylate it on one active site, bring it over to the other active site where pyruvate is, and then react the CO2 that is now activated with the pyruvate to make oxaloacetate. And so the CO2 is activated by being attached to the biotin, but the CO2 was also activated by ATP to get it onto the biotin. Two reactions make the oxaloacetate. This reaction actually occurs inside the mitochondria. And I make oxaloacetate. That oxaloacetate, we're looking at it from the perspective of gluconeogenesis, can go that way, but it also can, under extreme conditions, can be used in the citric acid cycle. Problem, oxaloacetate does not diffuse out of the mitochondria, so it has to be reduced to malate, and then the malate can be transported with an entrance of aspartate inside the mitochondria. Once it's outside, it was reduced. It now has to be oxidized back to oxaloacetate, and then we can do the second reaction of the bypass in the gluconeogenesis, and that is oxaloacetate to phosphoenol pyruvate. All right. During anaerobic exercise, you make lactate. Lactate stays in the muscle until it's dissolved in the blood flow, and then it gets into the bloodstream. Lactate will float around in the bloodstream, and it will be delivered to the liver. Liver actually takes up lactate. Lactate in the liver is reacted with NADH to make pyruvate. Okay? The pyruvate at this moment in time then can enter in the reaction that we just talked about and go through gluconeogenesis. And then glucose can get back in the bloodstream and go back to the muscle cell. This cycle right here has a name called the Cori cycle, and this is how we regenerate glucose usage. Lactate is a dead-end product in our biology in muscle. It has to go to a different tissue type, the liver, to be regenerated back into glucose. The oxaloacetate will react with GTP to create phosphoenol pyruvate. The power to run that is the loss of CO2. What happens is the enzyme facilitates the electrons to, sorry, that's the wrong place, form the CO2. The electrons form the double bond. This attacks the GTP at the alpha, beta, or gamma phosphate. Gamma, because we have a diphosphate as a leaving group. And we're now at phosphoenol pyruvate. All of the rest of the enzymes in gluconeogenesis are the exact enzymes of glycolysis. The equilibrium flows based upon which is the larger concentration. If the reactant becomes very large in concentration, it'll go towards product. Vice versa. If you have a lot of if you have a lot of reactant, it'll go towards product. All right. So we've learned the process. Now we have to turn it on and off. We've looked at glycolysis regulation, but we're going to look at it a little bit again in response to gluconeogenesis. There are two places where we really kind of concentrate on regulation. The conversion between fructose 6-phosphate and fructose 1,6-bisphosphate is the major control point in glycolysis and gluconeogenesis. Secondary is regulation between the pyruvate and the phosphoenol pyruvate. We said we had three enzymes that are bypassing, or three places. We do bypass up here in the glucose 6-phosphate to glucose, but it's regulated by another mechanism that we haven't really seen before, and we'll mention that when we get there. How we upregulate and downregulate the phosphofructokinase 1 and the fructose bisphosphatase 1. Okay, that is what's going to really make things run one direction or the other. This is the biggie. Either the presence or the absence of the 2,6 version. They are powerful allosteric regulators of both of those enzymes. And so, if you look up here in the case of glycolysis, with the addition of the 2,6-bisphosphate, it takes very little concentration of the reactant to get a really fast production of product. And so I will, with the 2,6-bisphosphate, I will make a lot of the fructose 1,6 with this molecule present. In the case of the gluconeogenesis, I will make a whole bunch of product, fructose 6-phosphate, in gluconeogenesis if I have an absence of the 2,6. So this is, and if you take a look at the top, 25 times higher for glycolysis if it's present, and 15 times higher for gluconeogenesis if it is absent. And so that is the huge, powerful regulator. You can see a inverse relationship between, is there a lot of energy or is there little energy? Okay, if there's not so much, if there's not a lot of AMP, okay, then I can make glucose back. If there is a a lot of AMP on this side and not a lot of ATP, I'm going to go through glycolysis and break it down. Citrates gets into this process. Citrate is a feedback mechanism for the pathway that is feeding it, that glycolysis feeds. If there's a lot of citrate, don't send things down to feed into the citric acid cycle. Okay? If there's a lot of citrate, hey, I can make glucose and I can regenerate it. If I lack citrate, then I'm going to feed the pyruvate, and then it will go into the citric acid cycle and go. And so we have outcomes of the next cycle, the fructose 2,6-bisphosphate, and energy charge that are powerful regulators. Okay, so how in the world do we control the 2,6 version? That is all hormonal. Glucagon, low energy state. Okay, if it is low energy state, I want glycolysis to go. And so the low energy state will cause protein kinase A to make this version. So low blood sugar gets us into the kinase, which makes 2,6, and when we have low blood glucose, we phosphorylate it, we make it the phosphatase, and what we're going to do is we're going to take the fructose 2,6 and make fructose 6, and if we have low blood glucose, that's good. What we want to do is we want to make gluconeogenesis go. The reason is we need glucose in the bloodstream for the brain. If we have lots and lots of insulin, high blood glucose, we have lots of starting material, it will cause it to be this state, which makes glycolysis run. And so if we have high sugar in the blood, we don't want to make sugar to feed it to the bloodstream. We want to use it up. If we have low blood glucose, like under starvation, the body will tend to start making glucose. And what happens is the cells start using fat and protein more because those are energy stores that it can use, and it will conserve and try to make sure that glucose keeps in the bloodstream just for the brain. And that's how it is controlled. Okay. The phosphofructokinase regulation, we've looked at this before, inhibited by protons, inhibited by citrate, and inhibited by ATP. We actually saw that on the last slide. We've actually seen this in glycolysis also. This is the last one. Glucose 6-phosphatase. This one's interesting. It is regulated by being compartmentalized. All of the glycolysis and gluconeogenesis happen in the cytosol. Okay? And the last step to actually make glucose itself, the glucose 6-phosphate actually has to be transported into the lumen of the ER. The glucose 6-phosphatase is working, and it will be only activated if the substrate is there. Once it's converted into glucose, glucose then can be transported out of the cell. We actually don't phosphorylate it. We don't do anything to it. We just put it inside a compartment to make sure that the glucose 6-phosphate is only deprotonated when we want to, because it's being pumped into that particular compartment. The energetics of gluconeogenesis, it takes 2 pyruvates, 4 ATP, 2 GTPs, 2 NADHs, which has the equivalent of 2.5 ATP each. So it takes 4 plus 2 is 6, plus another 5, which is 11 ATPs to make 1 glucose, the equivalent of 11 ATPs. The energetics for that is minus 9 kcals per mole. If I just reverse glycolysis, it's 20 kcals per mole. So I actually have to invest a lot of energy to make one glucose from the starting point of pyruvate. We do that to make sure the brain has glucose. All right. Let's take a look at the quiz really quickly. This is what I want you to do. You have 4, glucose 6-phosphates. Okay. The goal, make as much NADPH as absolutely possible. Okay. So, to make NADPH, you got to go through the oxidative step of that. All right. So, if I think about the oxidative step, all right, for each one, I also gave you a reference sheet. May or may not be helpful, depending upon how you view it. For each step of the pathway, for each one, okay, you're going to make 1 NADPH, you're going to make 2 NADPHs, and then you'll end up at the ribulose 5-phosphate, okay, and then we can convert those, all right? And so if I take 4, I'm going to do this. I will make 4 ribulose 5-phosphates, which can be converted into ribose 5-phosphate. So I'm going to say I'll get 5 ribose 5-phosphates. Okay, got 4 of those. I will each time lose a CO2. So I'll make 4 CO2s. Okay. I will also make how many NADPHs from those 4? 8. All right. That's the first step. Now, watch this. Glucose 6-phosphate. How many carbons are on glucose? 6. 6 carbons times 4 equals 24 carbons. Okay, that's important. Ribose 5-phosphate. How many carbons? 5 carbons times 4 equals 20 carbons. Where are my other carbons? CO2. There's 1 carbon for each times 4. I get 4 carbons. I have 24 on one side, 24 on the other. Don't ever lose these. Okay? You got to make sure each step has enough things. All right. Now, here you go in terms of the stoichiometry that you learned. If by chance, if you take 3 ribose 5-phosphates and you manipulate them, okay, you make a couple ribose and you can make a couple of them xyluloses, okay, then you can make the outcome 1 fructose 6-phosphate, 2 fructose 6-phosphates, and 1 glyceraldehyde 3-phosphate. Okay? And so, here's a way that you could do this. You have 4 of these. All right? Well, what you're going to do is this. You'll have 1 ribose 5-phosphate that doesn't react, and you have 4 ribose 5-phosphates that, oh, not 4, you'll have 3 that react. The reason why is because you got perfect stoichiometry with 3. Here I have 5 carbons. Here I have 5. I have 15. 15 carbons. Okay. And then, if you track it, all right, what I have made is 4 of these and 8 of those, okay? So I'm only going to deal with those 20 carbons in my next step, okay? So those 20 carbons are going to react here. All right. What I'll produce is, we said that we're going to have 2 fructose 6 phosphates, okay? And we're going to have 1 glyceraldehyde 3 phosphate, and 1 that didn't react, 1 ribose 5 phosphate that didn't react. And we still have the 4 CO2s that were produced, and we have 8 NADPHs. Now remember, I want to know as many NADPHs as you can make. Okay? I didn't create any NADPHs at that step. All right. What in the world can I do? Watch this. Okay? I can take this part and do a reaction. Everything else I'm going to drop down. 2 fructose 6 phosphates can become what? What can I convert glucose 6-phosphate into? Going through gluconeogenesis. Glucose 6-phosphate. So I can convert that into 2 glucose 6-phosphate. I didn't get any more NADPH. 12 carbons, 3 carbons, 5 carbons, 4 carbons. 12 plus 3 is 15, plus 5 is 20, plus 4 is 24. I still have all 24. I didn't make any NADPH. I want to make NADPH. Can you see something that will make NADPH? The glucose 6-phosphate. We can take those two. I'm going to react them. So I'm going to take 2 glucose 6-phosphates. And what does that create? Okay. At the end of the oxidative pathway, what did we make with each glucose 6-phosphate? Do you remember? Way up here. For each one, how many NADPHs did we make? 2. And what do we end up with? Ribose 5-phosphate. Okay, so check this out. These become 2 more ribose 5-phosphates. I already have 1, so now I have 3 ribose 5-phosphates. Okay, there's where the carbons go. I still have this 1. That didn't react. Okay, plus, in this, how many more CO2s did I produce? No, 2 more. Each one gives off a CO2. So 2 plus 4 is 6 CO2s. And then for each one of these, I can make 2 NADPHs, so I make 4 total, right? 4 plus 8 is 12 NADPH. Okay? 3 carbons, right? 15 carbons. 6 carbons. 6 plus 15, oh, no. Plus 15 is 18. 18 plus 6 is 24.
Is there a difference between ribose and ribose?
Yes. Okay, so ribulose can be converted into, these ribuloses can be converted into ribose or to xylulose, depending upon what you need. But remember, I told you that most of the time books say pentose phosphate pathway makes ribose. They can interchange between those through isomerizations and then epimerization. It can interconvert between those 3 things. Yes?
If we just see R5P, what do we assume it is?
Hmm?
If we just see R5P,
R5P will be the ribose 5-phosphate. Okay, I think they designate a ribulose as RB 5-phosphate. Okay. All right, I'm done. Yes. Okay. If by chance I have 3 riboses, I can go through this whole pathway perfectly stoichiometrically. What do I have? You've got 3 of those. Okay. Every time you think you're done, you will find things. So if you get 2 glyceraldehyde 3-phosphates, a glyceraldehyde can be converted into dihydroxyacetone phosphate. Those 2 can come together. Fructose 6-phosphate, glucose 6-phosphate, and then I can go one more time. And so every time you look at this, you need to find out where you end. You may even end up with a couple intermediates that are inside here that might react. And so even if you just have 2 riboses, okay, if you have 2 riboses, they can do this reaction and create a 7 and a 3, and then they can do a reaction that creates a 6 and a 4, and that 6 one can become what? Glucose, which they can go around again. Okay? Which then might give you another intermediate that might make another glucose. And so, there are multiple rounds to get to the final. Now, 3 would have worked out perfectly in a stoichiometry and you would have gotten everything converted perfectly. 6 would have worked out wonderfully. When I first gave this quiz, I gave 5 instead of 6. It took up like 3 pages, single-spaced typed to finish out the whole sequence. It was too much. 4 is still long, but it is like half of what it would be if I actually gave you 5 stoichiometrically. All right. What this does is it makes you think how you relate these structures in the pentose phosphate pathway and gluconeogenesis together and what could possibly happen. If you never did this quiz, you would never look at these structures really and understand what's going on. This is going to be complex. The worst case scenarios that I've seen in grading is people go on and 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 on, and by the end they have 48 carbons roaming around, and I only gave them 24. Don't make anything more than what you have, okay? So make sure you keep your count all the way through.